Thank you very much for the invite to the meeting. I've been asked to give you a talk on big data and personalized medicine. First of all, my disclosures, which are listed on the slide. We are now producing more data than we've ever done. We're producing approximately 5 exabytes of data every 2 days. That's 500 to 3,000 times all the content of the Library of Congress. If you do not know what that is, that equates to 5 times 10 to the 18 or 5 quintillion amounts of data produced every 2 days. Now, big data has been defined in various different ways, but there are 4 main elements to big data. First is volume, thousands of data points on millions of patients which are produced. Variety, data exists in many different formats, texts, quantities, images, biosignals, genetic sequences, etc. The data must also be accurate, veracity. Any biases and caused issues must be taken into account when undertaking analyses. And velocity. As I said, the data is accumulating rapidly from many patients and healthcare sites. A 5th V has also been added, that's value. People are trying to monetize data in order to be able to develop companies, sell data, etc. The UK medicines landscape is very much in keeping with big data approaches. The UK deals with over 1 million patients every 36 hours, 1.1 billion community prescriptions, and at a cost of £17 billion per annum. 8,000 hospital inpatients with adverse drug reactions at any one time, and 237 million medication errors. £300 million of money is wasted on medicines, and 50% of patients do not take medicines as intended. We're also facing a huge change in our demographics. The number of elderly people in our population is changing, increasing rapidly. 18% over the age of 65, and 2.4% are aged 85 and over. And this is going to change by 2035, as you can see on the graph. This undoubtedly leads to a patient population which has got multiple diseases, so-called multimorbidity, the presence of 2 or more chronic diseases, and those diseases need the treatment with medicines. And typically, polypharmacy is becoming more and more common. The number of people who are on multiple drugs, more than 5 medicines, has been increasing year on year over the last couple of decades. Now, in 2004, we published a paper in the British Medical Journal where we looked at 18,820 patients, and we found that 6.5% of all admissions to our hospitals were due to ADRs. We've just repeated that, a smaller version of the same kind of study, where we looked at 1,187 consecutive admissions to one hospital. Adverse drug reactions actually accounted for 16.5% of admissions this time. Those who in ADR are taking many more medicines, 10.5 versus 7.8, as you can see. Those who in ADR had more comorbidities, and the average length of stay for an ADR was 6 days. The cost extrapolated to the whole of the NHS England bed base is about £2.21 billion per year, a huge cost of adverse drug reactions. Apart from safety, there is also variability in drug efficacy. The vast majority of drugs, more than 90% of drugs we use, only work in about 30 to 50% of the people. Now, personalized medicine is a way of being able to improve the way we use medicines, and big data is going to be critical as part of that. There are many different definitions of personalized medicine and different terminologies as well: precision medicine, personalized medicine, etc. I'm going to use precision personalized medicine in the same vein. And basically, it is an emerging approach for disease treatment and prevention that takes into account individual variability in genetics, environment, and lifestyle for each person. And Obama used this particular definition in his talk a year or so before he finished as president. So what exactly is healthcare data? Well, it may be data that we use every day in our clinical care, such as electronic health record data, which may be in primary care or secondary care. Clearly, primary care data is much more readily available than secondary care data, given the multiple different types of electronic healthcare record systems present in secondary care organizations. Prescriptions clearly are an important source of data, healthcare data, laboratory data. We have huge amounts of laboratory data, very rich, but we hardly ever use that. Imaging data has been around for many, many years, but again, very rarely used at the moment, apart from for clinical diagnosis. There are obviously other databases as well, such as cancer registries, mortality data, etc., which are also important part of healthcare data. Other aspects which affect our health include climate change, the pollution in our atmosphere, and all of those are data which are available, which could be incorporated into healthcare data. Clearly, one of the major issues facing this country is the degree of health and race inequalities, which is related to deprivation, and we have data on that. And what we need to think about is that when we look at healthcare, we also need to consider social care data as well. These two are intertwined, and it's important to consider both together when we're looking at particular aspects. As patients are becoming more technologically aware, they're generating their own data. They can monitor illnesses using their mobile phones, and that data may be available. People are wearing wearable devices such as Apple Watch or Fitbits. Again, that collects a lot of data. There are sensors in many homes nowadays, and sensors that people wear. Again, that is collecting data. Many people monitor their diet. Again, that may be possible to collect and so on. And huge amounts of social media posts about illnesses, side effects, etc., which may also be useful sources of data to be able to understand the overall value of medicines. And clearly, patient-reported outcome measures are going to be important in the future. What do patients feel in terms of symptoms that they have, in terms of the drugs they receive, etc. And we need to be able to collect that systematically, incorporate that within what we do in terms of treating patients and using big data for personalized medicine. In addition to those kind of data sets I've already mentioned, there are many different omics technologies which are coming to the fore, which means that people are starting to use them and generating more types of data, which provide us much deeper understanding of what's going on in the human body in terms of physiology and pathology. Many of those are at the moment at an experimental stage, but genomics is clearly becoming more and more incorporated into healthcare, and is something that we need to be aware of. And I'm going to give you examples of how genomics is really important for what we do in terms of personalized medicine. Now, if you look at global genomics, there are a huge number of sequencing projects all going on all around the world. In the UK, obviously, you'll have heard of the Genomics England project, 100,000 Genomes Project. And in the United States, a precision medicine initiative that Obama started is now called All of Us. China's got a huge biobank. Japan has been undertaking a huge amount of sequencing, and many other countries are now undertaking sequencing projects, with data becoming available more and more frequently. The 100,000 Genomes Project actually finished in 2019. It was announced at the time of the London Olympics, and they were able to sequence 122,941 genes, 2/3 in rare diseases and 1/3 in cancer. What they've managed to find from that is that they've been able to identify cancer biomarkers, driver mutations, which allow potential for therapy in trials, but also 20–25% of rare disease cases have actionable findings and are able to get a diagnosis which they were never able to do before. Now, with so much data coming along, we need ways of being able to analyze it. Now, conventional statistical techniques are obviously still very widely used and are becoming better and better developed. However, artificial intelligence is an important area which will become more and more important as we move into the future, and will be required to be able to analyze the huge amounts of data that we're generating. Artificial intelligence is not actually new. It was a term which was coined by John McCarthy in 1956 in Stanford University. It's a subfield of computer science. Its goal is to enable the development of computers that are able to do things normally done by people, in particular things associated with people acting intelligently. However, there's quite a lot of hype about artificial intelligence as well. And it is really important that we understand the algorithms that are being developed by artificial intelligence, and we also understand the utility of those algorithms. And as Dr. Matthew Fenech said, that you wouldn't dream of prescribing a new drug without first having read the literature and being convinced that the benefits outweigh the risks for the patient in questions. Algorithms should be no different. So it is important that we do evaluate these AI algorithms as they come through. However, these are coming through. The FDA has started to approve quite a lot of AI algorithms. A list of those is shown here through up till September 2018. And we know that some of these are now being incorporated into clinical practice. For example, this particular deep neural network algorithm, it uses the iPhone or another smartphone to be able to look at particular dermatological skin lesions and is able to diagnose whether these are cancerous or non-cancerous to an accuracy which is similar to a board-certified dermatologist. Google DeepMind have been able to create an algorithm which allows prediction of future acute kidney injury, and this created a lot of controversy because of the use of data from one particular hospital in the UK. The area I particularly work in is called pharmacogenomics or pharmacogenetics. That's not a new term. It was coined by Friedrich Vogel in 1957, a study of variation in DNA sequence as a related drug response. In 1997, a Marshall, editor of Nature Biotechnology, coined it pharmacogenomics. And this is a broader term looking at the whole genome as well as RNA and DNA. And pharmacogenomic variants are very common in the population. This is data from 7.7 million US veterans. 99% carry at least one actionable pharmacogenomic variant. And 2.9 million of that 7.7, that's 37% of individuals, start a treatment with at least one level A drug. Level A means it's a gene-drug interaction where there's a high level of evidence from well-designed, well-conducted studies. Now, there is quite a lot of effort going on to be able to implement pharmacogenomics into healthcare systems, but most of the implementation efforts are basically scattered and are based on individual hospitals. For example, in the United States, there are some sentinel hospitals such as the Mayo in Boston, Chicago, where there are pharmacogenomic implementation projects going on. Qatar, for example, is looking at the possibility of pharmacogenomics implementation for its whole health system. That hasn't started yet, but that will be exciting when it does occur. The NHS has also been looking at this, and this will be the first large-scale whole population implementation. And we have the advantage that we have an integrated healthcare system to be able to do this. And we've been working together with the NHS to be able to look at implementation of pharmacogenetic testing in the NHS and the various different aspects one has to be able to look at. The first is the cost effectiveness. This is really important because if you're not cost effective in terms of what you're going to introduce, that will obviously lead to unnecessary waste of resources. And so work we've undertaken shows that implementing that kind of pharmacogenetic testing may actually be cost effective. The operational implementation becomes really important when you're trying to do this for 65 million population. What kind of test are the sort of labs going to use? Is it going to be one platform, many platforms, etc.? And that also has been looked at. Given that there are so many people on medicines, it is possible that the labs will become overwhelmed by pharmacogenomic test requests. Therefore, we try to create some eligibility criteria and also look at how one can report these tests as well. Importantly, for the future particularly, will be the development of decision support tools, which allow one to be able to incorporate pharmacogenomic variants into electronic health records and provide the prescriber with appropriate information and advice on what to do if somebody has pharmacogenomic variation. Clearly, anything that goes out from the NHS in terms of reporting, test results needs to be validated and needs a standard form of reporting. And those are particularly important areas in order to reduce errors that occur. Now, we undertook a workshop with NHS England, Genomics England, and this workshop has been published. And in this, we actually looked at all the opportunities and challenges for implementing pharmacogenomics for a whole healthcare system. These included things such as type of genetic testing, and what we came up with was saying that a phased approach was needed, which progressed from a panel approach, looking at multiple variants, to eventually whole genome sequencing. We've identified gene-drug pairs that could be actually looked at. We've also looked at the capacity for testing and developed those eligibility criteria. Clearly, turnaround time is important. You don't want to wait several weeks to get a result back so you can prescribe a drug. You want to prescribe sooner. So turnaround time is going to be really important. The clinical effectiveness and what is evidence-based that it actually improves things, again, has been looked at. Cost effectiveness has been looked at, and how to interpret pharmacogenomic results and advice, etc., has been looked at. But for the future, it's going to be really important to be able to train all the healthcare staff to improve their workforce skills so they can deal with pharmacogenomic testing. Clearly, it's also going to be important to be able to educate the public in terms of pharmacogenomics, its possibilities, but also its limitations as well. And it's going to be really important to be able to develop decision support systems. Good decision support systems are not quite there. We need to start off with basic decision support systems and build up capabilities over time. I'm going to show you one example of things which have been advancing, and this is in the area of serious adverse drug reactions. These are adverse drug reactions such as toxic epidermal necrolysis, drug-induced liver injury, and so on. And people have been particularly interested in looking for genetic biomarkers for these on the HLA complex, on short arm of chromosome 6. This is normally associated with the pathogenesis of autoimmune diseases, but it's also been involved in immune-mediated adverse drug reactions. And there are three areas of the HLA complex. That is class 1, which is A, B, C, class 2, HLA, DR, DQ, DP, and between class 1 and class 2 is the class 3 region, which is other immunomodulatory genes such as TNF-alpha, heat shock protein, and mica. A very good example of how genetics can actually help and prevent serious adverse drug reactions is with Abacavir, a drug which is used in HIV in combination with other drugs. Unfortunately, when it was first introduced, about 5 to 7% of individuals got serious hypersensitivity reactions characterized by skin rashes, gastrointestinal manifestations, respiratory manifestations, and some patients died on rechallenge. Work undertaken by Simon Malal in Australia was able to show a very strong association with HLA-B5701. This was replicated by GSK. It was GSK drug, and it was also replicated by ourselves. And what we also did was to show the cost effectiveness of this. What we showed was that by introducing HLA-B5701 genotyping would actually have saving of money in some cases, but it would certainly be highly cost effective. GSK, working with Simon Malal, went on to do a randomized controlled trial, which was able to show the B5701 pre-prescription genotyping was certainly clinically effective and cost effective. So in terms of implementation, we published this paper in 2004. By 2006, almost every HIV clinic in the UK was using HLA genetic testing before giving patients Abacavir. This is from 2006, even before the randomized control trial was published. The net effect of that has been dramatic. The Abacavir hypersensitivity has decreased from 5% to less than 1%, and I haven't seen a case of Abacavir hypersensitivity for the last 7 years or so. Since the beginning of the century, there's been quite a lot of studies to identify other HLA biomarkers for serious adverse drug reactions which affect the skin, like Abacavir, but also the liver, muscle, bone marrow, and so on. And these are all shown on this screen. For example, 5701 is associated with Abacavir hypersensitivity, but it also causes flucloxacillin drug-induced liver injury. Another very important HLA biomarker is HLA B1502, which was identified in Han Chinese individuals associated with carbamazepine-induced Stevens-Johnson syndrome. In Caucasians, HLA-3101 is more important and not HLA-B1502. We've developed a HLA gene panel, which is now been validated and CE marked, and it's available to the NHS. We were able to show the cost effectiveness of this panel for multiple pharmacogenes. We developed methods for estimating cost effectiveness, considering a multitude of effective drugs, clinical indication, and prescribing decisions. And for a £50 multi-gene panel test, looking at all these different genetic markers, we found it was both cost saving and more effective than standard care. In order to further implement pharmacogenetics, we've been undertaking a cluster randomized trial looking at a panel approach to be able to genotype individuals before they get drugs. This is part of a European project funded by the EU, and the aim of it was to implement preemptive pharmacogenomic testing in a real-world clinical setting across 7 sites. We're evaluating patient outcomes, cost effectiveness, using solid scientific methodology. We started this in actually 2016 by looking at developing enabling tools. The information technology tools that we needed to be able to really undertake this type of clinical trial, the pharmacogenomic infrastructure, what laboratory testing equipment was needed in the 7 laboratories which are undertaking this trial, the guidelines, what to do when you actually have a variant in your genome, and also training education associated with it. We've recruited 7,000 individuals which have been randomized to either standard of care or to genotype guided care. And what we're actually looking at in terms of patient benefit is a reduction in adverse drug reactions based on panel testing. We're also looking at the cost effectiveness, but also undertaking some science, looking at identification of rare variants, drug-drug interactions, and system pharmacology approaches. So the genotyping platform we used is shown on the screen here. It has 12 genes in there with 44 variants, which covers 39 drugs, which are widely used in the NHS. Every patient who has a genotyping test done gets a safety code, a pharmacogenomic card, which has a QR code, and you can link on with the QR code to particular data on the person's genetic profile, which allows you to be able to then determine what drugs the patients get based on the genotype. The sort of data from this particular randomized control trial is being analyzed and hopefully should be available later on this year. Now, one of the major barriers to implementation of pharmacogenomics in the population will be the lack of knowledge in our healthcare professionals. We've done work with pharmacists and with doctors, and the pharmacist survey that we undertook it showed that 66% felt knowledge of genetics was important, but 80% had little knowledge. 70% had no knowledge at all of what to do if a drug required genetic testing. When people were asked, would you like to learn more about pharmacogenomics? 42% put it down as a medium learning priority, while a minority said it was either high learning, or 8% said it was essential. Basically, they've got other things to learn and they've deprioritized this in terms of their learning. When we looked at doctors, overall knowledge is poor. Even when pharmacogenomics data is presented, some doctors ignore that pharmacogenomics data. The reason for ignoring was that they don't have time to go through the guidelines. And while pharmacogenomic testing is already going on in other parts of Europe and Australia, for example, it is common to be able to see this in pharmacists. It is not part of routine practice in the UK. So if we look forward to the future, not only is there pharmacogenomics, but many other technologies will become important as part of the personalized medicine landscape. And many of those different technologies are listed on this slide. And these were actually highlighted in this report, which is published by the Public Health Genomics Foundation. It was published on the 5th of September 2018, and the URL there shows you where it is, and you can download it to be able to read it. So let me finish. Data is reported to the main driver of the fourth industrial revolution. Data is and will increasingly be at the heart of healthcare. Big data and the huge amounts of data we're producing every day will require novel analytical approaches, including artificial intelligence, for us to be able to analyze it, extract insights from it, and to be able to put it into action that allows us to be able to improve patient care. Big data is certainly going to be a major driver of personalized medicine. And there are many different forms of healthcare data, and this will increase inevitably over the next few years, over the next decade or so. But we'll need to be able to integrate that into the way we treat patients. Genomics is already here. It's becoming mainstreamed in the NHS, and is being rapidly implemented in the NHS. The 100,000 Genomes Project was certainly an archetypal project which allowed us to be able to bring genomics into the NHS. Genomics and other big data approaches promise improvement in the way we use medicines, but many challenges remain. Increasing the skills of the workforce and changing practice will be critical, and that is one of the major challenges that does remain for us. Let me finish off by thanking all the individuals who have been involved in these studies that I've shown you, and thank you for your attention as well.